Hi, my name's Peter Canoll. I am a neuropathologist and a brain tumor researcher at Columbia University. I went to Brown University as an undergraduate. I graduated in 1987 from Brown. Decided while I was at Brown, I would do a PhD, which after graduating, I came to New York City, got a job at NYU as a research technician for about a year and for 2 years, and then at the end of those 2 years, I started an MD PhD program at NYU. I
Oh,
was
wow.
there until for a long time. From 1989 is when it started, and I graduated with my MD and PhD from NYU in 1998. My PhD work was on glial biology. I was interested in glial cells are one of the major cell types in the brain. Glial cells play a really important role in all kinds of neurological diseases. And in my studies, I started to pay attention to who were some of the leaders in the area of studying glial cells. And I realized that a lot of the people that were doing the kind of work that I was really interested in were pathologists, neuropathologists. So when I graduated from NYU, I did a pathology residency at Cornell Medical School in New York. And after my residency, I came to Columbia in 2000 and did a neuropathology fellowship. That ended in 2002, and I was hired here at Columbia as an assistant professor in pathology and cell biology, attending physician in neuropathology. I've been here ever since, building my research program, and I'm now the director of neuropathology. I run a fairly large NIH-funded research laboratory that is focused, as I said in the beginning, on brain tumors, but also other types of neurological diseases that involve interactions between glial cells and other cell types in the brain.
Could you talk a little bit more about what neuropathology is and
Yes.
the specific research you're doing in it?
Yes, sure. So neuropathology is a subspecialty of pathology. So pathology is a specialty in medicine, and pathologists are the people that surgical pathologists specifically are people that analyze tissue for diagnostic purposes, and neuropathologists are people who analyze brain tissue and spinal cord tissue and muscle and peripheral nerve to render diagnosis. So anytime someone has a biopsy or a tumor resection or dies of a neurological disease and has their brain removed at autopsy, it's a neuropathologist that analyzes that tissue, both histologically. We look at the tissue, by immunohistochemical stains and electron microscopy, but also now more and more by molecular and genetic analyses. And so the kind of bread and butter of a neuropathologist is to diagnose neurosurgical samples that are associated with brain tumors or epilepsy, for example, to give the patients a specific diagnosis associated with specific molecular diseases. So we work closely with neurosurgeons and neurologists as a team to help them understand the patient's disease so that they can optimize their treatment. It's a great subspecialty to be in if you're a physician scientist, as I am, because it's a laboratory-based medicine. And so it's very easy to bridge the gap between doing your research in a laboratory and doing your clinical work, which is also laboratory-based. And so when I'm doing my clinical diagnostic work, I'm also doing my research. A lot of my research is directly related to tissue-based analyses of patient samples. And I can go into a little more detail about that, but that's one big arm of my laboratory work. And another arm of my laboratory work deals with developing preclinical experimental models to study this, because there's only so much you can do when you're dealing with patients and patient samples. And there's questions that you want to develop an experimental system that will ask you questions that you can't ask in the patient samples. And so half of my lab is focused on studying patient samples and human tissue, and half of the lab is focused on developing preclinical studies, animal models and cell culture systems. And the two of them are kind of all focused in asking questions about the main theme of the research has to do with how different cell types in the brain and brain tumor microenvironment are interacting with each other. And to me, this is something that is relevant to all kinds of diseases, whether you're dealing with a neurological disease or a liver disease or a lung disease. But it's really important in neurological disease. Brain cells are evolutionarily evolved and engineered to talk to each other. That's what the brain cells do. They make contacts with many other cell types, and that's their speciality, if you will. And so those processes are hijacked or distorted during the disease progression, and they can either be disrupted or even contribute to the disease process. My interests in the research is understanding, I call them kind of like crosstalk between the different cell types, and understanding how those mechanisms contribute to disease progression, and also how there may be targets, therapeutic targets, ways of interrupting those crosstalks or enhancing those crosstalks in order to induce a better favorable clinical outcome. One way that we're studying that is through the analysis of human surgical samples to study what types of cells are there and how they're talking to each other in the clinical samples. So, for example, one type of experimental system we've developed is to take a patient's surgical resection. So we get a piece of brain tissue that the surgeon takes out of the OR, we bring it back to the lab, and we generate acute slices from the sample. So we cut it into 500 micron thick slices. So we have little slabs of the patient's brain tumor. We can make many such slabs out of a piece of tissue. And then we can treat those samples ex vivo, meaning out of the patient, with different drugs, different treatments. And then so we can have from the same patient, let's say we have 50 different samples, slices, and we treat each of those samples with a different condition, and then we'll do single cell RNA sequencing on the samples in order to understand how the treatment affected the different populations of cells and get an understanding of how a specific drug might have affected the neurons and the tumor cells and the astrocytes and the oligodendrocytes and the microglia. So that's one of the more exciting things we've been doing, and we've been able to take some of what we've learned from those slice cultures and bring it into clinical trial. Yeah, so one of the really exciting things about being a neuropathologist and being a researcher at Columbia is that you have the opportunity to work with a lot of different people. And I'm a big believer in team science. I think that if we collaborate, we can get more done. And so one of my very closest collaborators is a neurosurgeon named Jeff Bruce. He, for a long time, has been interested in something called convection enhanced delivery, where he understands, or as we all understand, that the blood-brain barrier is an obstacle to treating brain diseases, including brain tumors. So you can give a drug systemically, but only a little bit of it might get into the brain. And so it's hard to get the effects. So we have lots of drugs that can kill brain tumor cells in culture, but when you give it to the patients, it's not effective because it doesn't get into the brain. So Jeff Bruce's approach to this has been to put a catheter directly into the tumor and do something called convection enhanced delivery, where he infuses the drug directly into the tumor tissue, bypassing the blood-brain barrier. You can reach much higher levels, but also you avoid the systemic toxicity, because even though you have high levels of the drug in the tissue, you don't achieve high levels in the system. And so if there's toxicity to the liver or to the kidneys, you avoid that with the local delivery. And what he's been able to do, because he's bringing the patients in and surgically implanting the catheter, at the time of the catheter placement, he takes biopsies of the tissue and then delivers the drug into the tissue, puts a implantable pump, refillable implantable pump into the patient's abdomen. And so the patient can go home and they'll have this pump delivering the drug. And then after the time of the drug delivery, which in the last clinical trial lasted a month, they'll bring the patient back in and he'll remove the catheter and then take more biopsies. So we have tissue immediately before and immediately after treatment, and we can do the same type of analysis that we're doing on the slice cultures to see if we're targeting the same types of populations, which cells were affected, which cells were escaping the therapy. And through that approach, have been able to understand not only how the drug treatments are working, but in what ways they're not accomplishing what we want to accomplish. So, for example, in this last study, we were able to show that the drug we were giving, which is a drug called topotecan, was able to kill all the proliferating tumor cells, but it didn't kill the non-proliferating tumor cells. So the cells that were not killed were left behind, and those are the cells that eventually will give rise to recurrence. And so that has directed us towards a lot of efforts to try to identify ways to target those non-proliferating quiescent, we call them, tumor cells. And so that's led to a big effort where we're using the slice culture study to test out different drugs that might target the non-proliferating tumor cells. And we've identified some really exciting candidates that we're exploring.
I know that the field is expanding a lot, and even just in recent years, technology has been absolutely incredible in the way that it's expanded. How have you used that in your research? And like, where do you see the field of neuropathology going?
Yes, so that's a great question. Thank you. So molecular genetic analysis is a major part of neuropathology right now. So for every patient who has a brain tumor surgery at Columbia, and also at most major medical institutions, the sample is sequenced. So when we make a diagnosis for a brain tumor, we make it based on both the histology, meaning what we see in the tissue, what it looks like, and also the molecular features. It's called an integrated diagnosis. And so that is the standard of care now for brain tumors. But it also is the leading edge for research because we now understand so much about the molecular makeup. We can start to design treatments, and there's multiple levels of molecular characterization. One is to understand how the DNA is mutated, to understand the actual mutations that are driving the tumor. And sometimes those represent potential therapeutic targets. And they also represent good ways to classify the tumors. So we classify them based on the genetic alterations, but they don't always make good therapeutic targets. And so a different type of analysis is called transcriptional analysis, where we're doing sequencing of the genes that are being expressed into mRNA. And that analysis has been done now for over 10 years. We've been performing RNA sequencing on large numbers of brain tumors to understand what we call their transcriptional phenotypes and to classify the tumors into different classes of disease based on which genes are upregulated or downregulated at this expression level. But now, over the last 5 years, I would say we've taken that a step further, and now we're taking the tissue and we're dissociating it into individual cells, and we're sequencing, getting transcriptional sequences of each one of the cells. It's called single cell sequencing. And now that means from 10 years ago, from a patient's tumor, we would get the RNA sequencing and the genomic sequencing of that patient's tumor. One genomic sequencing, one RNA sequencing. Now we're dividing up into thousands of cells. We're generating thousands of RNA sequencing libraries. And so each tumor, we're not only getting the molecular alterations of the entire tumor, but also of the individual cells. And I would say that that's no longer the future, that's the present. That's what's happening. Molecular analysis, that's one of the exciting things about pathology. It's really the leading edge where research and molecular diagnostics are brought into the standard of care. So it's what I do as a pathologist, is that kind of analysis. There's another area that's kind of just taking off, and I'm super excited by it. And I think in the next 5 and 10 years, you will see this blow up. And that is digital pathology. So when we get histologic sections of a patient's brain tumor, and we stain it with different things like hematoxin, and each of these, so a tissue section, microslide, with a piece of tissue on it, stained with something, and it will give a pattern. And we as pathologists look at that under the microscope and we interpret it based on our diagnostic skills. Oh, that looks like a tumor, or that looks like normal brain. But what we're doing now is we're digitizing those, and we're training computers through machine learning and artificial intelligence approaches to interpret it. Radiologists have been doing this for longer than pathologists have, and have developed really advanced tools to interpret things like MRIs and X-rays. And it's just a form of image analysis. And there's all kinds of machine learning tools that are very good at being taught how to recognize patterns. And so this is kind of what I would say is the emerging future of pathology, to combine the digital pathology and image analysis with the molecular pathology, putting those kinds of data sets together to inform machine learning programs to help with diagnosing, identifying potential therapeutic targets, assessing response to treatment. These are the kinds of things that I think are just coming online as part of patient care. And neuropathology is a really great area to explore that in, because we're doing it on every single patient at this time.
Yeah, I have noticed that I think AI is bleeding a little bit into every aspect of research and even other parts of my life. And then I want to backtrack
Yes.
a little bit and ask how did you get your start in research, and how do you suggest high schoolers begin their journey in research?
I didn't get interested in research myself in high school, and I really applaud high school students that are interested in research. I think that's amazing. And if you are able to see how wonderful an activity it is and how rewarding it is at that young age, then cheers to you. I always enjoyed science classes for as classes, but the endeavor of research, there's science, learning what's known, and that's great. And I think that's probably the inroad that all students should take. If you enjoy biology, physics, chemistry, any math, computer science, psychology, any of the sciences, if you find yourself fascinated by that material, then you should try out research. And research is a different activity. It's going into the weeds a little bit. It's asking something that isn't known and test it. It's really, I get one of my favorite part of my jobs is I get to mentor students. And when they come to the lab, I meet with them for the first time, and I tell them about what we're doing, like we're talking about right now. And then I try to transition as soon as possible into figuring out what interests them most in what we talked about, and get them started with the process of asking questions, which is when you're doing research. If you could go into a lab and do an experiment that someone designed for you, and that can be very exciting and rewarding, and that's how it should start out. But then when you do an experiment that you designed yourself, to ask a question that you came up with, that's when the magic happens. That's when you see them all of a sudden like become totally engrossed in it. And it's both, it's both it's like a labor of love. It can be very frustrating. You can run into brick walls. You get lost in what you're doing. And so it's become the research activity becomes this kind of like walking the tightrope between the known and the unknown, and asking questions that are answerable, and figuring out how to do an experiment where whether your hypothesis is right or wrong, you're going to learn something and move the bar forward. And so it really is it's a really unique experience. And it's interesting to be a physician scientist, because practicing medicine is in some ways the exact opposite of doing research. When you're practicing medicine, you're exercising a known activity where you have the high confidence. It's all about experience. I've seen this before. I know how to do this. Patients come to you because you know how to do what you need to. In pathology, people rely on my expertise because I know how to do something that I've done many, many times before. And then in research, you're doing something you've never done before, and you're asking a question that in the unknown. And so you have to have a completely different mindset. It's wonderful to see students wrestle with that and come up with their own solutions of how they're going to handle the uncertainty of research, kind of like figure it out, and whether they even like it or not. It's not uncommon that people will do research and decide that it's not for them. I've seen a lot of medical students, for example, do research. They do research really well. They're able to do the experiments, ask the questions, interpret them. At the end of their research year, they'll be like, well, that was great. I'm never doing that again, because I'm going to go do surgery. I want to do heart transplants because I want to be the expert that knows how to do what I'm doing. Very often the other way happens too, where someone gets really excited about a question and they fall in love with it, and it becomes their obsession to be a researcher.
You mentioned how you take in students. Theoretically, if you were to hire a high schooler, what type of traits are you looking for in someone?
What a high school student, I've had a few high school students come into the lab. And
Oh,
so right
wow.
away, the fact that they're even interested in coming to a lab as a high school student right away is amazing to me. Because not only was I a high school student, but I also have had 3 high school student children of my own, and I know high school students, and so I kind of awe of a high school student that wants to spend, let's say, the summer in a research lab. What I would hope, I don't expect anything a priori from them. They've already kind of like, the fact that they show up kind of like earns them the credit of being, of giving the opportunity to learn something. What I try to help them achieve is the challenge that we have to overcome is we have to give them a project where they are going to learn something. They're going to be mentored. It requires that they work, a high school student or a college student, or even quite frankly, a graduate student. Early on, you need to have mentoring. You need to be working very closely with someone. So, we're talking about things like being respectful of other people's time. When someone says to show up and do something, you show up and you do it. If you do those things and you're pleasant to be with, you show interest and enthusiasm, you might, that's what it requires, really. And then you might show an aptitude, if you're open-minded, you take feedback well. If someone makes a suggestion, you consider it and you try to take their suggestion and act on it. You have follow through. You do what you say you're going to do. These are the things. So basically, that's the same kind of approach to a research as you would do in any job or any starting positions, right? You go in there, you be pleasant, you interact with the people, you listen to what they're doing, you try to take their advice. And then in your free time, if you're really fascinated by something, you can do your own research. And what I find is like when you get like to the A++ kind of levels, if someone comes in and they say, I've had high school students say, I was reading this paper last night and I had this question that I'd like you to ask you about. And it'd be like, wow, this high school student went out and read a paper and was able to formulate a question that they want to ask about. I mean, to me, that's everything.
But yeah, that's all the questions I had for today. Thank you so much. I really enjoyed talking to you and listening to everything you had to say.
I enjoyed it as well.